Hi everybody and welcome to our methods presentation. Our group was chosen to do the pulse chase experiment. My name is Gwyneth Stubbs.
I'm Jackie Peterson.
And I'm Fernando Gill.
All right, and then on to the next slide. The purpose of this method is to analyze the cellular processes that occur over time. Additionally, it can provide important information such as the half-life of molecules and measuring DNA production. In the field of cell biology, this method has been utilized in important studies, such as George Palada's discovery of the order in which proteins move from one organelle to another within the secretory pathway. On to the next one. The pulse chase method is divided into 3 different stages, the first being the pulse phase. The pulse phase occurs when cells are exposed to a large amount of a radio-labeled compound over a brief period of time. In this picture to the left, a radioactive amino acid, tyrosine, is introduced. The labeled amino acid is then incorporated into the proteins via the process of protein synthesis, which can be seen in the picture. The red dots on the finished proteins represent the radio-labeled tyrosine. On to the next. Following the pulse phase is the chase phase. In this stage of the method, the unincorporated radio-labeled compounds are washed away, and the cells are exposed to a large amount of the same compound that is not radioactively labeled. In this picture, it is normal tyrosine. This compound is incorporated just like the one in the previous step. On to the next slide. The final step is periodic sample testing, which involves taking samples throughout the chase phase of the method to analyze the location and chemical form of the radio-labeled compound. By taking samples, the location of the compound introduced in the pulse phase can be traced over time. In the picture on the left, the radioactive labeled proteins travel from the rough ER to the Golgi and finally to vesicles that will transport them outside of the cells. To the next slide.
For the analogy I chose for the pulse chase experiment, I chose the catch and release tagging experiment of a model species of organism. Let the pulse phase represent the labeling or tagging of the species of interest. Next. Then, during the chase phase, after all the organisms of interest are labeled, you allow the organisms to go to their downstream environment. Let the cell be represented by the upstream environment, and the downward streams represent later life stages within the organism. Next slide. Next. Back. As you can see in the chase phase, we monitor to see the rate of the life cycle within different parts of the stream or different parts of the cell. Then you can find other information about the organism or track it through the chase phase. Next slide. For our article of interest, we chose the pulse chase method being used in an experiment with Alzheimer's. Next. So amyloid beta peptides are indicative of early stage Alzheimer's. It's due to presumably an imbalanced rate of synthesis, folding, and degradation. It ultimately leads to the formation of plaques, which are required for Alzheimer's, but not alone sufficient for enough formation of plaque to cause this later symptoms. Next. The amyloid precursor protein, or APP, is a transmembrane protein of interest that's also thought to be involved in the formation of plaque and later AD manifestation. Seeing as it is cleaved to form AD peptides and is involved in the synthesis of amyloid beta or degradation of amyloid beta, it would make sense that they would conflict with one another. So for the hypothesis of the experiment, next, the scientists were going over the assumption or hypothesis that hampered amyloid precursor protein, APP, turnover contributes to AD pathogenesis. So APP mutations of interest, such as genotypes, human APP mutations that were expressed in the genotypes of the mouse model species at knock-in contribute to the accumulation of the beta proteins and plaques. And then questions based off of this hypothesis are, where is APP protein turnover hampered most within the cell, and where does the synaptic dysfunction originate? So tracking synaptic dysfunction throughout the location of the cell. Next slide.
So here we cover the methods of how pulse chase was used to reveal the location of the synaptic dysfunction within the brain. So on this figure on the right, we can see how the mice were actually radioactively labeled with nitrogen-15, and by being fed nitrogen-15 labeled chow. So when they breed, they went through the F0 and F1 generations, and eventually they took tissue samples from each of their brains, comparing the pathogenic genotypes, the APNLF and APNLG, with the control genotype, APNL. And to determine the places where protein turnover was being hampered in the cell, they used mass spectrometry and computer programs to calculate fractional abundance of nitrogen-14 and nitrogen-15 peptides. So for each protein, the nitrogen-14 and nitrogen-15 chromatograms for the peptides were averaged, and the protein remaining was calculated from those averages to compare the genotypes. So on the second part, right below where it shows the mice and the N15 pups, it shows how quantitative mass spectrometry based proteomics was used to do this protein turnover analysis. So for their results, they used the multi-isotope imaging mass spectrometry, also called MIMS, which measures these ratios of nitrogen-15 to nitrogen-14 in the brain tissue sections, which allows you to visualize which macromolecules contain that radioactively labeled nitrogen-15. So in figures 5G and 5H, you can see these are the MIMS results, and the N with the lines is pointing to where those were localized. And another way they probed for this nitrogen-15 signal is by using antibodies for this amyloid beta plaque, which is one of the precursors to Alzheimer's disease, and the VAMP-I, to investigate if the signal colocalized with the plaques or hampered presynaptic proteins. And they show that in figure 5I. And in both of these figures, you can see that they were localized. So they found, and this is their results, that the APNLG tissue sections had nitrogen-15 signals, which co-localize with VAMP-I and amyloid plaques in the presynaptic terminal, suggesting that these amyloid beta aggregates can sequester these presynaptic proteins. So another interpretation, an easier way to understand this, is that the presynaptic proteins have elevated levels at early stages of alpha amyloid beta accumulation. And the axon terminals, the presynaptic terminals, are the selective sites of impaired protein degradation in these pathogenic genotypes of the APNLG and APNLF brains. So that is the results of this experiment, and that is pulse chase. Thank you very much.